Sorry, is it recording? Okay, yes. So today, as told you, so we are gonna study about this paper recently published in Nature Material 2025. Title is Cytoplasmic Anillin and Act to Promote Low-Wave Myosin 2 Dependent Confined Migration and Invasion. So first, when you look at the paper, you can see received data is November 2023. And then accepted May, published June. So it took around 1.5 years to publish. So if you publish in a good journal, it take long time. Yeah, you should remember that. So, and then one thing is that if you see some unpublished data in the conference, and that data will be published like 1 or 2 years later. And then they already start the experiment 1 or 2 years before. So most of the good study, they are designed in 4 years ago from the data of the accepted. So you should remember that. So they designed this experiment 4 years ago, and then they perform like 2 years of experiment, and then 1.5 years revision, and then finally publish in Nature Materials. And then, so probably you will know about the cytoplasmic. Cytoplasmic is that cell, there's nucleus, like brain, and another part is called cytoplasmic. And Anillin Act 2, you never heard before. I'll explain it. The low-wave myosin 2 dependent. Probably some of them you are familiar about the low-wave myosin 2. Low-wave is like, ectomyosin contractility. And in the cell, when the cell move their body, their morphology, they have such skeleton like actin, microtubule, intermediate filament. But those kind of skeleton, they cannot move by themselves. They need some special muscle-like structure, which is called myosin 2. Okay? The low wave is the upstream of myosin 2 to activate the myosin 2 activity. Okay. And then depend on confined migration. Confined migration is when you see the cell on the cell culture plate, they are naturally spread out, right? Just, but confined means that they should penetrate very narrow and then narrow site. So they should, when in the cell point of view, in the body, they want to move one place to the other place, they cannot fly. They have to penetrate some extracellular matrix, which means that they have to penetrate very small cytopore, sometimes 10 micrometer, sometimes 2 micrometers, something like that. So cell, their average diameter is around like 20 or 30 micrometer. So when they penetrate very small pore, they should be confined. Confined means that they should be like squeezed. So this is the meaning of the confinement. And then migration invasion. Migration is movement from A to B place. Invasion is from cancer, from cancer point of view, when cancer invades, migrate to another normal tissue, which is called invasion. So technically, as a meaning, migration and invasion will be very similar. But migration is like normal cell migration, and invasion means from the cancer cell. Okay, let's see what's the meaning of this one by one. So, okay, first, let's start about the cytoplasmic nucleus cytoplasmic. Let's assume that you are very junior and then you never heard about this. And then like this, first, see the title and then see what's the meaning of that title keyword. So this is okay. And then you can see from the picture. So first of thing that you should see their meaning definition in the dictionary and then see what's going on in the picture. When this is a cell, this nucleus is cytoplasm. And this is a cell membrane. And probably nucleus cell has membrane, so it outside of the layer is nucleus membrane here. Okay. Now we come to know that what is cytoplasm here. What is the other thing? Other thing is Anillin Act 2 is a little difficult, so let's see later. Low-wave myosin 2. Okay, low-wave myosin 2, copy and paste. Okay. So definition here from the AI. Nowadays, Google show that low margin tail pathway is crucial signaling cascade in cell where the small duty page low activate low kinase, which in turn phosphorylates numbers margin 2. It's very difficult. So anyhow, there are some cascade or pathway. And when you look at the images like this, this is the good things. Very small. Okay. So when you look at this one, you see that? So when something happens, so low way activate rock, and then rock make phosphorylation of MSC, and then phosphorylation MSC activate myosin 2. Yeah. So this kind of cascade is low myosin 2 pathway. It's a basic pathway to make the force. So in terms of a cell point of view, when cell move or when cell like transfer something inside of their body, they need this power. So this power is most of them coming from the low myosin 2 activity, especially actin. Okay. So as you know that microtubule, they have another like methodology to move the molecule like dynein or say the other protein. They have another linker to move the protein through the microtubule. But in terms of actin, they should be driven by this low myosin 2 activity pathway. Okay. The next one is okay, migration invasion. Okay, maybe probably you know about this. Let's see Anillin Act 2. Actually, we have to remember and then imagine how they focus on this Anillin in Act 2. Okay. Before that, let's see about the confined migration. Maybe you guys never heard about confined migration before. So let's see. Confined migration is this one. Hmm. Yeah, this is a good example. Okay, let's say this is an epithelial cell, and then maybe let's say this is a cancer cell. Most of the cancer cell, they're maybe 90% of cancer cell is coming from epithelial cell. They should be lying in most outer layer, like skin. But something happened to them, then they have to migrate through cell-to-cell interaction and then go inside of the dermis layer. Dermis layers means that extracellular matrix of the skin. And then when they penetrate this small pore, small site, the cell should be squeezed like this. And then after penetrating this epithelial cell, and then after they're reaching this ECM, also they have to penetrate this collagen or ECM like fiber structure in 3D, right? So in any point, when cell migrate to A to B site, they should be squeezed. Not all the time, but most of the time they should be squeezed. Okay. So when you culture the cell on tissue culture plate, and then we are doing a lot of experiment like, okay, tissue culture plate, put the cell, and then when cell move A to B, but there's no confined, right? Only very free area. They can go anywhere. There's no external force from the ECM. But when you culture the cell in hydrogel or certain kind of like membrane which have pore, and then when the cell migrate up to down, they should penetrate the pore. So basic method to check the confined migration is to use insert. So insert is very easy. You can buy and then see the cell on top of the insert, and then you can check how many cells can go down, how fast. This is one of the way to check the confined migration easily in 2D. And then in 3D, you should check some collagen structure, collagen gel, or any kind of scaffold like that. So I can say that confined migration happen a lot. Yeah, but most of the time, as a biometric scientist, you are familiar with the just migration study on tissue culture plate or material on the material. But in reality, cell most of the time cell migrate in confined area. And when they confine area, so they need some power to squeeze their own body. Can you imagine which kind of cell component is the most rigid component? Definitely nucleus, right? So they should squeeze the nucleus. And then where is that power from? This is their basic question. Okay. And then from the many like pathway, they are focusing on this anilin act 2. So anilin act 2, for me even, I never heard it before. So I know act 2 is cytokinase. This one? Mm. So when you say easily was that cell division, but in this paper, cell division is academic terminology. They call it cytokinesis. Cytokinesis is a little very similar to cytokine release, something like that, but they are not related to cytokine release. You know that cytokine release, like cytokine 폭풍, something like that, right? So cytokine, the cell, they can release cytokine easily, like interleukin beta, TNF alpha, growth factor. But this cytokinesis is like cell division. It's a very different terminology. And then when cytokinesis happen, already mentioned about that. When cells are dividing to two sites, two daughter cell, they also have to cut down the nucleus, right? And then when you imagine that, they need some power to cut, right? Okay, this is a nucleus and cell. Even though you wanna cut in half, you need the power to cut using the knife, right? But from the cell, how they make those kind of power? So for that, they are using this Act2 and anilin structure to slice the nucleus. So the author, they just found how they generate this much power to squeeze their nucleus. And then they just check probably this Act2 anilin could be involved. Because from the biology point of view, this is the most powerful generator to cut down the nucleus. And when they cut down nucleus, probably they can squeeze nucleus very easily, right? So this is their rationale, how they focus on this. Okay, let's see. And then this Constantopoulos. Yeah, probably this guy is coming from Greek. Yeah. So when I search it, he got the bachelor degree in Greek and then went to USA for PhD and then they're doing research in Johns Hopkins Medical School now. Mm. Okay, let's see. So figure one. So first, they have to let cell migrate, right? So they are using three different confined area. This is like cross section, okay? So, but when you see the in 3D, maybe this kind of hole, they are squeezed out, squeezed. They are go and they squeezed inside. But there, this cross section area is 100 micrometer square. It's like 10 multiplied 10. 30 is 10 multiplied 3. 9 is 3 multiplied 3. This is the most confined microenvironment, while 100 is almost less confined, right? So going down, this more confined area from left to right, all the paper, this is the migration of direction, okay? So when you see, and then they are cultured the human, oh, what is that? Some cancer cell, fibrosarcoma. Oh. So when I said most of the cancer cells are coming from epithelial cell, but non-epithelial cell cancer cell is called sarcoma. Sarcoma is like 살코기, something like that. Okay. So when they are migrate non-confined 1 micrometer square area, this is kind of structure you can see. This green color is actin. When you see 30 and 9, blab. Blab is kind of like this is your intact cell membrane. But sometimes cell membrane is like curvature like this. This is called blab. Yeah. So you can see this is blab. And then this is also blab. So from the 30 micrometer square, they can see the blab. So they count blabbing cell 100 micrometer, just 10%. But 9 micrometer, most of the cells are blabbed. Blab means normally when you see the blabbing, which is a whole marker of apoptosis, cell death, or when cells get a lot of stress, they're blab, like external force or chemically. So blab normally is a whole marker of stress. And then when they're stressed, okay, we already know that actually this confined migration was published in 2005 in Science, something like that, by the Lemerding, who is a professor in Yale University. So after that, they come to know that this when the cells squeeze and com- they can migrate in confined area, they already know that they have to generate certain power, which is low wave activity. So this signal lifetime is low wave activity. Low wave activity is not easy to detect. So because of that, they are using very special dye, flip flap dye. Then when the meaning of this one is that lifetime when they're decreasing, which is a high activity of the low wave. So in lab imaging, they found out compared to 100 micrometer scale, 9 micrometer scale cell, they have less lifetime, which means more low wave activity. Definitely, okay? When they squeeze out, they need some power, low activity, okay? And then they quantify in very detail, like rare part, perinuclear part, front part. So this is what I want to highlight you guys. Most of the time, okay, for C, you have to do a lot of experiment, live imaging. You got this data, okay, but this is not the end of things. Because definitely when you see this structure, maybe 1 micrometer, probably similar, but 30, can you see that? Where is more different color? Back side, front side, and near nucleus, right? Here also, back side, near nucleus, front side. So when you see the data, and then, maybe I can stand average of this, and probably I can also quantify this spatially, back, center, front. Okay? So you try the basic thing, a basic difference between, let's say, IT members and the other mechanobiology scientists. Other mechanobiology scientists, they try to quantify everything. Like, okay, I want to quantify back, front, center, and then maybe top, upper, something like that. And then you can get a lot of data from the one experiment. Okay. So you try to think about how you quantify again and again, more in detail. So rare perinuclear front, most of the things they are similarly. 9 micrometer have less lifetime, which means they have more lower activity. They are generating more power. And then, okay, this power, low activity is upstream, right? And then low rock, MLC to positive MLC, and then myosin 2 activity increase. So downstream, which kind of myosin 2 is activated? A, B, C. SH is like silencing. They try to silencing A, B, C, A, B, A and B. They found out that A, when they're silencing, blabbing cell go down, which means that they are experience less power, right? And then we can say that not B, but A is involved. We can say that myosin 2 is involved because 9 micrometer here, 90% of cells should be blabbed. But when they low down the myosin 2A activity, their blabbing cell number is decreasing, which means that blab 2 is something happening. They are involved in this phenomenon. Okay. The cell entry. So they didn't say anything about this. Probably we can imagine, all the cell can go inside, but not really. When they count cell entry percentage, and then we can say that, not all, but some of the cell, they can migrate to confined area. So definitely 1 micrometer, 60% more, 9 micrometer, 10% less. But Y27-6 is a very famous low activity inhibitor, as you already know that. So when they inhibit low activity, because they found that low activity is enhanced. So they block the low activity by inhibitor. They found the entry number is decreasing. And that cell entry time, how long does it take to when the cell they initially search out the entry site and then they touch and then okay, I can go there. And then they try to like up, go and back, and then finally they can go. This duration is called cell entry time. How long does it take to go inside of that pore? So when they check the time, 1 micrometer maybe less than 50 minutes. And then let's change of by low activity inhibitor. But when you look at 9 micrometer, oh, it took around 100 minutes. When they block low activity, it took a lot of time, 2 times. So this or figure one, what is called confinement induced low activation of two facilitated entry and bleb based migration. Okay. So one thing I want to share with you is that figure one is very important, and then you should try to position the figure one very good data because okay, as a reviewer, as an editor, when they evaluate the papers or your manuscript quality, most of the time they focus on title, abstract, figure one. When they're not interested in reject. Okay. So after doing all the experiment, you have to think about how I can highlight my experiment, how I can make my data fancy or cool. Mm. So this is relatively cool, I think. Mm. And then mechanism study, you can present later. Mechanism study, we should do that, but it's a little boring, right? Sometimes boring. So, and then mechanism, they just, okay, as I told you, they just shotgun anilin first because anilin is the most well-known protein to cut down the nucleus in cytokinesis, cell division. So in 2D, like tissue culture plate, anilin normally located in nucleus. But when cell is divided, this anilin go outside of nucleus and they cut the nucleus and they go back. So that's why in 2D, most of the anilin is detected inside of nucleus. It's line to peak. But 1 micrometer also all of them nucleus. But from 30 micrometer square, what happened? Anilin is located in front, back, and front. Definitely we can say this is like near the cell membrane, edge, right? The 9 micrometer square, there's no anilin in the nucleus. And you can detect in back and front, right? This is very obvious data. Oh, and then the hypothesis is proven by this experiment. Oh, and then they call this kind of anilin location front and back is called AC. This is like what is that? Their terminology AC is anilin reaching at edges. Okay, I forgot it. This one. Anilin Richie, Jones, and set edges. They just call it like very fancy aces. But Anilin Richie, Jones, and set edges, ACE, aces. So they quantify how many of aces, my god, aces is detected. Aces is okay. You can see that this is a little this is aces, right? Little thin here. So maybe 50% aces you can see. But over the decreasing of the pore size, ACCs come to almost 100%. And then we can imagine the ACCs could be important to regulate this kind of confined migration. And then they found out that, okay, this annealing is really related to low wave activity or some actin. So they culture two different cells, H2, HT is our original cell, fibrosarcoma, MDMB231 is a breast cancer cell. From two cancer cell line, and then they magnify these images, they make the line here. They found out that anilin, red color, and green color, low A, red color, followed in actin, blue color is well localized. it edge when they're on 2D structure. Edges like this, here, here, and here. So they can say that probably in locational point of view, this annealing low wave actin, they are co-localized together. So that's why. And then they check this annealing is really overall for all kind of cancer cell or any kind of cell. Annealing is all detected. Not only this HT, MD, MB, another cell line. So its annealing is a very well known protein. And then they hypothesize that, okay, probably this annealing is key things. And then they want to know that how this annealing is leaked out from the nucleus. As I told you, most of the annealing is localized in nucleus. But some special occasion, like when cell is divided, cell squeeze like this, they could be leaked outside of nucleus and they are deposited front and back. So, and then from the previous paper, they mentioned about that nucleus itself, they can act as a sensor. So when you imagine, maybe this is, let's say this according to this scale bar 10 micrometer, this 10 micrometer, and then probably the nuclear size around 15, something like that. When they squeeze in 3 micrometer, from 15 to 3 micrometer, 5 times they should be squeezed, right? And then as a previous paper in Science publishing, nuclear itself, they can act as a mechanosensor. So for proving the concept, they change their cell cycle to enlarge the cell and nuclear size. From the same cell, how could you enhance the nuclear size? Most easiest way is to change osmotic pressure. When you treat the cell like DW, 33%, their cell volume is enhanced around 20%. And then probably nucleus is also enhanced volume. And then one thing easily you can see that when you change osmotic pressure by dW, adding the dW in media, you can easily change it. But somehow here they are using another methodology to increase the nucleus. How? They change cell cycle. They stop cell division. And then most of the cell, not all, most of the cell is like, G0, G1, S, and then they are divided. G0, G1, S, G2, and divided. So at G2 level, they just stop the cell cycle. And then most of the cell is occupied in G0, G1, and S. So then S means that synthesis. So when, when nucleus, they're making more DNA for cell division, En come to be 4N, right? And then at certain time, they are divided En, En. Sorry, they're divided 2N, 2N, right? So extended GNS means that they are enhancing DNA amount to increase the nuclear volume. And so therefore, as a proof of concept, they check cell nucleus area, aspect ratio, and the volume is supplementary, they are enhanced, but not 2 times like that, just few. But still, and they parallelly investigate what's going on. So first, okay, let's forget about the extended G1S, but let's focus on G1S. Green color is anilin GFP. This MNS M cherry, NS is like nuclear. When you see some NLS in certain paper, which means that protein, when they have NLS binding site, which means that they can go inside of nucleus. NLS is detected by importer protein. Importer protein is delivery guy, which deliver the protein from cytoplasm to nucleus. For recognizing which kind of protein can be delivered inside of nucleus by importing, it is detected by NNS domain. This is a peptide domain. So this domain, M cherry. So this NNS protein, and then dyed by M cherry red color. And then this is like detecting uh, how much of protein they, they originally should be inside of nucleus can stay inside or outside the nucleus. It's like we can easily say that nucleus protein overall. Okay, MS M cherry. GF anilin is anilin protein. Definitely GF anilin also has NNS. That is why they are localized in nucleus originally. But as you imagine, more the protein, they should be fabricate a synthesis outside of nucleus, and then they can go inside, right? So when you imagine like DNA, RNA protein, DNA is inside of nucleus. When the DNA is transcribed to RNA, they can go outside of the nucleus in the ER. Their RNA is translated to the protein, and then they are released. And then finally, they can go inside the nucleus, or they can maintain in cytoplasm, or they can go outside of cell, right? So all the nucleus, all the protein, when they are designed to go in the nucleus, they should have NNS. So and they check red one here, red most of nucleus, but somehow, let's say this 1 hour later, some this not very strong color but dim NNS, they're leaked. And then as you can see, these edges, anilin, they are detected here, 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 here. And then this is from the back, little front, back front, back front, back, little front. Okay, this has happened over time. Not only GNS, extend GNS. And but important thing is that this extend GNS, they have more saturated color, which means that their protein amount in nucleus is like larger than normal cell because they are extended. Some cell they are making duplicated DNA. That's why they have more NSM cherry, so that we can easily see the leaking of this nuclear protein to cytoplasm. But similarly, you can see that nucleus protein leaked outside of cytoplasm. And then this anilin also you can see front, back, and front, back, and front. And when they analyze frequency, like frequency, they quantify from the many cell. So nowadays, people want to write down their n number inside of the figure or figure legend at least, like this, n number, something like that. And then they check rare AC. Rare means that backside. This is the backside. Front is front side. Rare this ace first, or front AC first, or front and rear same time, no ace. Quantify based on this image, lab images. And the frequency, NER is like nuclear envelope rupture. Envelope means nuclear membrane rupture. How they make the rupture? This is no rupture, right? But from here, little dim red color, you can see that. Can you see little dim color? So they measure the time. This little dim color when they are leaked out. But this leak out is first, or ACE develop is first. They said ACE develop only, and then but at the moment no leakage of this NNS around 80%. And then at the same time, very few. But this ACE phenomenon after NER 20%. So this kind of quantification, when you live imaging this one, and then you have to think about what's the best quantification to this, and you try to quantify like this. Frequency, and then NER first or ACE first. So why they are checking this NER and ACE? So why they are doing this experiment? They want to know which one is first, which one is later. From this experiment, previous experiment, they found out in confined space, anilin is detected, backside, front side. We want to know where it's coming from, right? This is our basic question. For answering that, probably this anilin could be coming from original nucleus. So, but interesting is that because from the previous images, we can imagine that here, let's say, oh, where is that? figure 2. Here, okay, we can easily see when you look at this 30 micrometer compared to 1D micrometer dim, right? But you can see ACE here, and the 9 micrometer, nothing in nucleus. But cytoplasm, back and front, you can see that. And then we can easily imagine, oh, this annealing and this ACE structure, annealing back and forth, is coming from the nucleus, right? But probably as end point, most of them is coming from nucleus. But here, they find out initial time. Initially, this is like some endpoint, but initially, let's see, this 40 minutes later, anyway, we can see this kind of A structure. Even this A structure, where is coming from? For answering that, they quantify like this. And then from this one, no rupture or age detected, which means that probably they have already some anilin is already localized in cytoplasm, maybe 10% of total. And then when they squeeze out, somehow this ACE is developed first. And then over time, the cell migrate in small area, they need more power. So from the nucleus, this anilin is leaked out, and then they are deposited front and back. They describe like this. Mm. And then actually, why they're doing this extended G1S? They want to know nucleus can act as a mechanosensor. That's why they check first nuclear rupture after entry to channel, which means okay, when you see that, when how long does it take to have this NS leaking from cytoplasm? Here, relatively no leaking, T0. Rupture 1 means that they are little leaked, little color here, right? Mm. This is little color like this. So from based on this, they can say, oh, how long does it take? This time, extended GONS less, 0.05 below, which means they are fast. They are fastly making nuclear rupture. Why? Their nuclear size is already a little bigger than normal. And then rupture per hour, how many times nucleus is ruptured? Maybe normal cell 1 or 2 times, but extend more bigger size of nucleus 2 times, something like that. So from this one, they can say any nuclear sensing could be involved for making this kind of nuclear rupture. And then GFP annealing, this A structure also. And then for linking this nuclear rupture, and then annealing, start of annealing nuclear exit, they correlate this phenomenon and they find out extended GNS more fastly they are making start of annealing nuclear exit. How they measure? So this annealing is most of the time they are inside nucleus, but from maybe from here they are little leak out like that. So they quantify this how many times their nuclear exit. But this red color one, they are more fastly go down. Their combining one is 1.0, 100%. When the frequency is more deposit in less time, which means that this annealing is more fastly going out in large size of nucleus. Mm. Actually, from my understanding, this is not that much important. Yeah, nucleus, everybody knows about the nucleus can act, they can sense some mechanical force. But anyhow, maybe probably some reviewer ask a lot of these questions, so they are doing like this. So as aniline edge structure exists in cell on 2 dimensions and are amplified by confinement. 2 dimension means that tissue culture plate, and the confined structure also there located, localized. And then they found out, okay, this phenomenon is we all the time we are culture the cell on like glass because when you or PDMS, when you make this confined area, you have to use some micro fluid chips When you attach microfluidic chips in certain cover slip, they need certain substrate, right? But they want to know that when they change the substrate stiffness, or when they culture the cell in 2D and 3D hydrogen structure, or fast relaxing hygienate, what will happen? Similarly happen or not? Because they want to generalize their concept. So here, 21 kilopascal, 2D culture plate, they're like this, 39 micrometer, also detected A structure, right? 8 kilopascal, or little dim in 30, but 9 very dominantly you can see it, A structure. So they found out that annealing intensity is higher, rare nucleus front in high stiffness. Okay, which means that when even though they are migrate in certain area, when general stiffness is enhanced, the ACE is more deposit. Can you imagine why? Because they need more power. They need more power to migrate. So when you go this way to the other way, when this matrix is very soft, you need more power. But that generally when stiff is very when this matrix is very stiff, you need more power to pull and push. So definitely from that national, they hypothesize higher stiffness need more power. They prove it from normalized annealing intensity, and then from 30 micrometer square, and then 9 microsquare stimulus. And local rupture, definitely rupture is more enhanced in 21 kilopascal. Because anyhow, when they squeeze out to higher stiffness substrate, they have to generate more power, and then this power can make the rupture of nucleus. And not only these all experiments, they can do, like say, 2D. 2D means that PDMS substrate and then chip on top, and then this hole are they're migrating. But when they culture the cell in 3D hydrogel, and then maybe collagen concentration 2.5 microgram per ml, collagen also very like they are mingle each other very very localizedly. So when they migrate cell in 3D structure, they need very much power. So, and they check 2D, 3D from this collagen, and then 2D, 3D with GM. GM means that MMP inhibitor. Okay, when they migrate somewhere, let's say 3D structure, when your cell, you imagine your cell, you are occupied by a lot of like collagen like this. You have to like that, put and push one by one. And the other way is that when you can secrete some MMP, it's like degradable enzyme, you can degrade chemically collagen. And then when they degrade it, and then you can go easily, right? So the meaning of this GM MMP inhibitor is that they block this chemical degradation, which means that they have to penetrate, migrate physically without any help of biochemical factor. Okay, and then definitely this with GM is a more harsh condition for confining, right? So when you look at this, uh, especially this 3D NT and then GM, you can see when you look at this and that, also 2D, you can see this is NNS more leaked out, and then GFP aniline little leaked, but not that much from a confined area, right? This is without this is 2D means that without chips, only collagen on top. That is why there's no A structure, no like this structure here. But you look at the 3D normal condition, there's no NNS leak out, aniline leak out. But when they block MMP, now they have to physically migrate in confined area. You can see this NNS is leaked out here, and then A structure is detected. That they found out that, oh, this is really happening phenomenon in 3D collagen hydrogel, right? So they tried from the beginning of the microchip fluid study, and then now it's collagen, that they try to generalize this concept. Not only your platform, but also in reality, it can happen. Later, this paper also they mentioned about the in vivo tissue as well. So when you find certain biological phenomenon, first as a model, you can show your material, something like that, and then over time your material, general material, or a general in vivo tissue, something like that, to generalize this concept. And then here they are finding fast relaxing arginate. Probably this collagen also relaxing gel, but this also some people are asking. That's why arginate, not only collagen, but also they synthesize arginate gel they are making, and then they found out that this 3D structure also you can see more annealing is detected edge when they are migrating in 3D structure. So they found out that migration in biomimetic 3D environment promotes annealing recruitment to ACEs. Okay. In the figure 4, now in vivo tissue. Okay. So how they do that? This their model cell is HT something. This is fibrosarcoma. So they die fibrosarcoma using this. They die like NNS cherry for dying nucleus, GF anilin, sustain anilin, SSG is like collagen structure in vivo tissue, like skin. The dextran is like blood vessel. So they inject their cancer cell in nude mouse skin. And then they check. So so this A is like non-confined one. And then S annealing like that. When you look at the right side, there are a lot of blood vessel, SSG. This SSG like this straight, this one is coming from the muscle. So when the cancer cell is localized, this is also collagen fiber, but maybe this is like 10 micrometer, but this is like 20 or 40 like that. But here, especially this when cancer cell is squeezed instead of this 50 micrometer, 40 or 6 or 10, even the 6, when you highlight it, you can see anilin is localized in edge or front, like this. But and the anilin is a little leaked out. So they compare this leaking is coming from 6 micrometer, but not 40 micrometer. 40 micrometer represents the image is coming from A, no annealing, H, and then it's leaked. Here, red color a little bit out, and then this blue color up and down, right? So for in vivo tissue, also you can quantify like this. Now and then from this side, there tumor core and invasion zone. When you inject the cancer cell in certain area, definitely you can see this part is core. Some cells are there migrating to other surrounding normal tissue. This is called invasion zone. Invasion zone is more like they get more stress. They're more squeezed. So invasion zone percentage of ACEs larger than the tumor core. Here, this their quantification. See, this 20 micrometer probably is like 5 micrometer confined area. And the NLS leaked out, annealing detected front and back. And even this SSG is like collagen structure or muscle structure, dextran is blood vessel. So this vessel near area, this cancer cell is squeezed. So the one thing is that cancer cell, when they migrate from this side to other side, one of the easiest way is to use the vessel, blood vessel, right? They go inside the blood vessel. Oh, they go inside the blood vessel, and then they can flow out to other side, right? So this cancer cell somehow they are inside the blood vessel color they are co-localized. When they come inside the nucleus, and then they flow out. But this vessel is very small diameter, 5 micrometer. So they should be squeezed like this. Okay. And then when you look at the other side, this is another like dexterous vessel. near vessel, a lot of like muscle or collagen inside the vessel. Also, they can see that aniline, the A structure, NS is a little linked, but actually this DNF very similar. But DNF, their quantification from E and G, but as far as understanding, they said F is like non-NS leaking. Even though you can see this one, but this is like live imaging. So when you live imaging in vivo tissue, you can, after making defect on skin, and then you can suture some glass structure with the link, metal link, and then for a certain hour, you can visualize the cell inside of the skin using two photon confocal machine. So this is like live imaging. So from live imaging, this NNS, when they leak, they are leaking, they are leaking from the beginning. Beginning, there's nothing, they are leaked. But this NNS white color is like their artifact somehow. But here is upper one, they are really leaked out from the movie. When you look at the movie, you can see that they're leaked out. So they want to say that when NNS is leaked, well, without leaking, this aniline structure, A structure, you can detect. Mm. Now this quantification is very not easy to follow, but one thing you can see that this NNS NC control, just control group, no squeezing cell. NC upper one is like squeezing cell. NS NC, this one, and then anilin, this M is membrane, C cytoplasm. Membrane is more edge site. Is anilin A structure is going up like this from this dot going up, and then NS will go down, up, down. This is not very 100% make sure, but they show the tendency. Little up while they're going down. But from the NNS leaking, or here NNS is even go up or very similar. But this aniline also very similarly detected. So without leaking, with leaking, without leaking, this aniline A structure can be detected inside of in vivo tissue. Why they mentioned that? Initially, they mentioned without aniline leaking, aniline originally some part they are localized in cytoplasm, and initially, aniline is localized in edge, and over time, when they squeeze out, more aniline is leaked out from nucleus, more deposit, right? So they have to prove both concepts. Original aniline in cytoplasm deposit in edge, plus over time, in the squeeze out, aniline is leaked out, and then there are more deposit. Because of that, they have to prove both concepts. But as you can see, it's not easy to say this is really happening. But anyhow, they're trying to show this tendency. But from the movie, you can definitely see that. And then we already show our concept depending on the confined area when they are more squeezed out from small size, more A structure they are detected, right? For in vivo tissue also, constriction width we are decreasing, decrease constriction width decreasing. Oh, yes, decreasing, more A's percentage detected, right? Makes sense, right? Concentrate with less is less confined, more confined. So they prove their concept in vivo tissue. So other figure 5 and 6 I will do Thursday. So today we are gonna taste this paper. So I want you to guys read at least twice of this paper to next lecture Thursday, and then let's we can go other things for Thursday. So do you guys have any question up to this? Should I ask any question? Hmm? Yeah, right. So now you know some concept here. So now it's easy to read the paper. Mm. And then as a backup, nowadays, if you want to publish in Nature material like that, let's say we have six main figure and then extended figure, very similar quality of the main figure, but not that much important data in extended figure. So nowadays, maybe main figure, figure 1 and 6, extended figure, figure 1 and 8, and supplementary figure 1 and 10, total figure 30, something like that. This is the concept of donation material paper. So when you can see that, you can see, okay, many cell, similarly blebing cell, their quantification, perinuclear front layer side, and then other 100 micrometer, no change by normal smashing 2. But here, another cell line, they can see that it is like actin also, you can bleb here along with the NLS. So they check all kind of like cancer cell, and then most of them anilin they detected. And then actually Jeffrey anilin. So when you say Jeffrey anilin, NLS, and cherry, this like externally you adding this protein, which means that they are not endogenous. Endogenous is natural. Endogenous anilin is a natural anilin, but it's not easy to detect lively. That is why you are there are some plasmid which can generate anilin instead of nucleus. But this anilin is specially fused with GFP, so you can visualize where is anilin. But people say that, oh, when you add anilin more, this is like non-natural cancer cell. You add overexpressed anilin more. It can be same or not, right? They can ask this question. That's why they detect definitely endogenous anilin compared to that one. When you add GF anilin, you can see more nucleus anilin. But for other figure, you can they said endogenous anilin, GF anilin, their tendency is very similar by staining. I'll show you. So here they said this is like anti-anilin. This is without GF anilin. They just stain them. No transfection. Even anilin staining, they can detect 30 micrometer channel, they're more squeezed out, right? So wild type cell staining, GFP anilin, and then you stain with anti-anilin. And the GFP anilin, and then you can detect by the GFP color. All of them, they are similar tendency. This one, and then one the microchannel, GFP, sorry, this is like just normal cell and anilin staining. They added GFP anilin and they overexpress anilin, and then they stain them and then check by GFP. Very similar they detect, right? And then this is like prove their concept. There are two GFP anilin is worse. You can when you overexpress this GFP anilin, they are similarly happening. This phenomenon. Not only their HT cell, breast cancer cell, other cancer cell, you can see most of the rare nuclear front. This 30 micrometer confined one, they have more anilin intensity, the edge. This is another cancer cell like that. And then, actually, this is very important data. We have to say that this anilin is really combined with the low A. So I'm going to show you the mechanism later. But for answering that, this is like, okay, total cell lysis, total cell. Nuclear extract. From the cell, you only extract nucleus. From nucleus, you can detect the anilin. From cytoplasm also, you can differentiate nucleus at the cytoplasm in certain way very easily for Western blot. So they want to say that total cytoplasm nucleus, they can detect anilin. Because normal people, they said anilin should be inside the nucleus. But they want to say anilin also localized in cytoplasm. And they control JP trap IP. And then from this JP trap IP, which means that JP trap anilin, from the anilin, you can detect the low A because all these experiments, they want to say that anilin could be kind of sponge scaffold to sequester the low A. This is their concept. So for proving that, they show the Westerblot data here. Yeah, well, this is a very important data. linking to the next lecture. Okay. So I want you to guys read this paper and it's 2 times, and then let's see Thursday. Okay. Thank you.